pioneer, Genentec Incorporated. He also served as a consultant to help biotech giant Amgen redesign the training programs for their biomanufacturing technicians and for their quality assurance associates. He currently serves on the advisory committee for the Explorer Education Division of Biorad, a biotech firm that provides equipment and materials to schools worldwide. He's also on the advisory committees of several colleges and high school biotech programs. In 2000, the Association of Community College Trustees chose Jim as the distinguished faculty member of the Pacific region, which includes the Western United States and Canada, Hawaii, Alaska, and Guam. In 2011, Solano College faculty chose Jim as the distinguished faculty member of the year. In 2015, he was a finalist for the Award of Excellence for Faculty Innovation, the American Association of Community Colleges. I give you Jim DeKloe.
Thanks, Dave. Thanks. And sometimes individuals aren't appreciated in their own area, but you should know that Dave Menchu is nationally famous, and everyone knows about Modesto, and everyone knows about the excellent science education that you have here. Dave travels all around the country, and he and I hang out in, last time was New Orleans, and before that it was Houston. So thanks very much for the introduction, Dave. Yeah. And we've known each other for decades, and we've had a close collaboration. Thank you very much for inviting me. I spent a part of my youth in the Central Valley, in Tulare to be specific, and I'm always happy to come home in a way. I am going to spend tonight trying to convince you that my field of biotechnology is the most important field in the world. And I recognize that I have a stake in it. I recognize that I have a bias. But I am going to make the case for you that this is the field of the future. And let me bring in some experts. Here is Freeman Dyson, the late Freeman Dyson, a famous physicist. You may have heard of a Dyson sphere. He, later in life, was talking about biology. And in a book, he said the following: the 21st century was the century of physics. The 20th century was the century of physics. The 21st century will be the century of biology. Biology is now bigger than physics as measured by the size of the budgets, the size of the workforce, and the output of major discoveries. It is likely that biotechnology will dominate our lives and our economic activities in the second half of the 21st century, the way that computer technology dominated our lives and our economy in the second half of the 20th. That is, in biotechnology, we're going to see the equivalent of an internet. The fundamental change caused by an internet. Here are some others. The fourth industrial revolution, categorized as evolving the fusion of physical, digital, and biological technologies, will likely be fueled by operationalizing engineering, biology, research, and manufacturing. A fourth industrial revolution. Here are two reports, and both of them say the same thing, that the bioeconomy writ large will be worth $30 trillion as a global economy. $30 trillion by the end of this decade. Here again, the fourth industrial revolution. Synthetic biology is about to disrupt your industry. Again, another consultant coming up with the same number, $30 trillion. Now, you may have noticed that in September, President Biden signed an executive order that was going to devote quite a bit of the recently passed CHIPS and Science Bill to the building of infrastructure for biotechnology. Again, here's this number, the equivalent of almost $30 trillion in terms of value. Now, to put it in perspective, Brian Deese, the director of the National Economic Council, said in the summit that accompanied the executive order, that's a really big number. Currently, the GDP of the United States is $21 trillion. You're talking about 50% more in the single industry, 50% more than the current entire economic output of the United States, all goods and services sold and performed in the United States. And he turned to his people, he said, I had them check the numbers, that's the number. So transformation, advances in biological science could help transform economies and societies, helping to tackle global challenges from climate change to pandemics. And we just lived through a triumph of biotechnology. I'm on many national committees. Some of the people on those national committees are from Pfizer. I have to tell you that they're taking a victory lap. They're, we did it. So if you think about it, the first reports of this strange respiratory illness coming out of China was December 2019. By January 11th, 2020, the genome of this unique RNA virus was sequenced and published. Pfizer got busy, and they started talking about it, but they got busy in earnest in March 2020, and they scaled up manufacturing. They got clinical trials under Operation Warp Speed, and they got not full approval, but emergency use authorization by the end of the year. No one's ever done that. And that is a triumph of biotechnology. There are many, many other vaccine stories, but that is one good example where a vaccine was produced in record time, really in a large extent. I have to be frank with you, I thought we were going to have a depression, not a recession. I thought that we were going to have a 1930s style depression, and we didn't. And part of the answer for that was the speed and efficacy of the vaccine. Folks in the back, come on in. We're friendly if you want to. Go ahead and sit down. I won't be offended. Good, and let me put it in historical context. So let's pretend that you're living here in Modesto in 1900. What's your life expectancy? Well, half of you aren't here. And college students, you're looking a little old because you don't have much time left. Infant mortality, most of you have lost half of your children. Really common. If you look at historical figures, Abraham Lincoln had 4 sons, lost 3 out of 4. Thomas Jefferson lost his first wife to infectious disease. He married a widow named Martha, lost 3 out of 5 children. George Washington was sterile. He encountered a fever starting the French and Indian War. He almost single-handedly started. But he married a wealthy widow whose husband had died of infectious disease, and she had 4 children, had lost 4 out of 5. It didn't matter if you were rich or poor, you would lose 4 out of 5. Now, the good idea, or the good trend is life expectancy now is in the 80s. Infant mortality, you expect your children to live. Now, I would like to make the argument that constitutes a fundamental change in the human condition. From going from knowing for sure that you're going to lose half of your children to expecting your children to live. And there was a medieval practice, and if you're a Game of Thrones fan, George R.R. Martin adopted this for North of the Wall. There's a medieval practice, you didn't name your children until they turned 2. Don't get too attached, you're going to lose most of them. Of course you did get attached. The frequency of death did not lessen the devastating impact of losing a child. Charles Darwin had 10 children, lost 4, including his favorite Annie. When Annie died, he locked himself in his room, and his wife said, "Look, you've got other children. You ought to come down." Yeah, I know. I just can't. I can't. So, what made this fundamental change in the human condition? Biotechnology. Okay, so vaccines, of course, and many of the vaccines that children take are directed against horrendous diseases. The number one killer of children used to be diphtheria, where a pseudomembrane forms in the throat and the child asphyxiates. My father-in-law recently passed. He was a retired pediatrician. He trained in medical school in the 1950s. They had whole wards of pertussis, whole wards bigger than this room of children gasping for breath. They had whole wards of measles, of children that had the encephalitis associated with measles. You don't even see measles anymore. A friend of mine is a radiologist in Napa. One time they thought this kid had Hodgkin's lymphoma. They thought they had a form of cancer. And he put up the X-ray, and he said, this is weird, that salivary gland, he's got mumps. No one had seen mumps. They didn't even think of mumps. He has mumps. He runs down the hall. He has mumps. They didn't even think of it. No physician, because of the vaccines. Antibiotics, of course, probably half of you are alive because sometime in your life you had a bacterial infection and it was treated with antibiotics. Don't neglect nutrition, especially vitamins and knowledge of that. All through the South, there was pellagra. It was a nutritional deficiency. Also, all through the South was hookworm, and all through the South was malaria. There's a reason why the Centers for Disease Control and Prevention is in Atlanta, Georgia, because malaria all through the South, that was the old malaria lab. And then don't neglect sanitation. It is always bad to drink fecally flavored water. And that isn't because of the icky effect, because many, many diseases are transmitted by a fecal oral route. And so all of this knowledge has been garnered over the years to increase life expectancy, diminish infant mortality, and we're now on the next phase. We're at the brink of multiple revolutions that I'd like to talk about today. Now, bioscience has a role for everyone. Biotechnology, everyone says, oh, that sounds so science-y. There's a lot of roles that are less science-y. So not only scientists, but healthcare workers, technicians, quality assurance, information technology, business and supply chain. There's a lot of paperwork if you're making pharmaceutical biotechnology products. You have to comply with the Food and Drug Administration regulations. People that are very good at reading and writing, and that's for them, regulatory compliance, intellectual property. And so I've gotten to it. What is biotech anyway? The broadest definition is biotechnology is the use of living organisms to produce value-added products. So by this definition, what neighbor Gallo does is biotechnology. Gallo takes grape juice, which a bottle is worth maybe $1, and then puts in a yeast, a living cell, a unicellular fungus. That unicellular fungus takes the sugar in the grape juice and converts it to alcohol and a little bit of CO2 that you usually vent off or you may capture in champagne. And then you slap a label on it and you sell it for tens of dollars. Or if you're in Napa, you put a fancy French name on it. Cabernet Sauvignon, Sauvignon Blanc, Pinot Noir. You put a really fancy label on it and sell that for $150 a bottle. So who did the work? The yeast. Who gets the money? You do, ka-ching. So biotechnology, it's adding value. You take a raw material that isn't worth much money, add a living cell, and convert it to a product that's worth a lot of money. And so here's old biotechnology, vinegar, bread, of course. In bread, you capture the CO2, and that's what causes the bread to rise. And then you bake off that ethanol, and that's part of the heavenly aroma of baking bread. Cheese, of course, the cultured milk uses bacteria, and so those are old biotechnology. Currently, biotechnology is the production of medicines. The use of genetically engineered organisms to produce pharmaceutically active protein. But at its core, it is the same process. It is alchemy. The old alchemists, they dreamed of turning a base metal, lead, which it wasn't worth much money, into gold. This takes a giant tank filled with sugar. You add a genetically engineered cell. It takes that sugar and converts it into protein that may be worth 200,000 times its weight in gold. And by the way, that protein is addressing unmet medical needs. What that means is that protein is going to be used to address a disease for which there's no treatment. Let me say that again. Not for which there's no cure, but addressing diseases for which there is no treatment, for which medical science has no solution at this moment. So there are many, many parents out there with children with genetic disorders. They're watching the scientific journals, hoping that a breakthrough in this industry will come in time for their son or daughter. So one of the products of biotechnology is hope. So where I live in Vacaville, California, we have Genentec Vacaville. I'll show you a picture of the plant. Genentec Vacaville was built for this product, Herceptin. This is an antibody that's directed against a protein that is especially abundant on the surface of an especially aggressive type of breast cancer. Before this product, a woman who was diagnosed with HER2 positive breast cancer, their physician would sit them down and say, get your affairs in order. You will not survive the year. After Herceptin, those women are surviving beyond the 5-year mark. Here is my favorite drug. That's tissue plasminogen activator. I'll tell you about it later, but it's an enzyme that digests blood clots. Why is that useful? The most common cause of death in the United States is a heart attack. The most common type of heart attack is a myocardial infarction. It's a blood clot in a coronary artery. That blockage blocks blood from flowing, and heart tissue downstream of the blockage begins to die. You inject this enzyme, it finds the clot and digests it, restoring blood flow. Not very different than putting Drano in your sink with that hairball and having the Drano digest the hair and allowing your sink to drain. Likewise, it's very useful for treating strokes because the #1 cause of stroke is a blood clot in an artery that is feeding blood to the brain. If that blood clot is allowed to continue, all the brain tissue downstream of that blood clot begins to die, and the patient will lose the function of that part of the brain. Again, you inject it, and it goes to the blood clot, restores blood flow, and there's no loss of function. How much is that worth to you, do you think? Is it worth $22,000 a gram? Yes, it is. So remember, you're going to hear about high cost of medicines, and you're going to hear, especially, you're going to hear it less now that the elections are over and all of that. It's an easy target. But remember, if an ultra expensive drug saves a single night in the hospital, it's probably paid for itself. That is, the cost of hospitalization is so high. It's just that these drugs are very visible in there. So it isn't only the science, which we tend to focus on, but it's the engineering part of it, making the machines that do this, the piece of equipment that do this, medicine, and equally important is the business side. I've seen many biotech companies that had excellent science, but the business didn't come through, and therefore they ran out of money and they had to close their doors. The business side has to come through, and also the government side, especially regulation by the Food and Drug Administration, a branch of the executive branch, a part of the executive branch of the United States government. So this began in the 70s, so the early people all wore leather vests and had long sideburns and long hair. But this began with genetic engineering of cells, the ability to make recombinant DNA molecules. And here is the city of South San Francisco. They rightfully claim that they are the birthplace of biotechnology. That is, this field, which is now all over the world, is a California story. And we still, in the state of California, dominate the field of biotechnology, and we will continue to dominate. Why? Well, remember in the 1950s, we had a visionary state government that put in infrastructure, put a lot of emphasis on education, the community college system, the Cal State system, the University of California system, and also put a lot of money into infrastructure, Highway 5 and the California Aqueduct, so Southern California can steal our water. They put a lot of money in, and that echoed in especially aerospace, in computers, and in biotechnology. And again, the San Francisco Bay Area continues to dominate in biotechnology. In this one city, South San Francisco, if you've ever been there, on the top of Mount San Bruno, it says the industrial city. They were a city that had a steel industry, but steel moved overseas and they were looking for a new industry. So they invited in the first biotech companies with inexpensive land, and we'll come back to that. And the science came out of the labs also, especially Stanford University, University of California, San Francisco. University of California, San Francisco, scientist Herb Boyer was at a conference in Hawaii, and sitting in the audience was Stanford professor Stanley Cohen. And Stanley Cohen said, I've got an idea, and he invited him out to dinner, and they went to a delicatessen. And over a delicatessen in 1973, they thought of this. They thought, if you had an enzyme that you have, Herb Boyer, and you cut DNA with it, and you cut another piece of DNA with the same enzyme, you can stick it in and get a bacterial cell to have any gene, including a human gene. So then Herb Boyer got a call from this young entrepreneur, an unemployed recent graduate of the MIT Business School named Bob Swanson. And he said, Bob Swanson said, have you ever thought of commercializing your technology? I have access to money. And Herb Boyer said, access to money? My lab always needs money. And he said, I have contacts with venture capitalists. And Herb Boyer said, what's venture capitalist? And so, yeah, I'll meet you for a beer, and I'll give you 15 minutes. And that 15 minutes turned into 3 hours, and by the end of the time, they were writing on the back of an envelope, genetic engineering technology, Genentech. And that meeting over a beer is immortalized in bronze on the Genentech main campus in South San Francisco. And I've been told that if you want your experiments to work, rather than throwing a coin in the fountain, you have to put a coin in Herb Boyer's beer mug. So, and then they had to go get money. Well, first they borrowed some money to hire a lawyer for articles of incorporation, and then they went to Tom Perkins. And Kleiner and Perkins was one of the early venture capitalists, and they had this model of venture capital that they had used to build early Silicon Valley. Before venture capitalists, if you wanted to start a business, you need some capital, you need some money to hire some people and to rent a space, and you'd go to a bank, and the bank would say, what's your collateral? I don't know, I can put my house up for mortgage, but that isn't much. What's your collateral? I don't have any collateral, but I just have a good idea. And so what came out of Silicon Valley is this concept, this approach of, I will give you money for a piece of your company because I'm taking this huge risk. If it pays off, I'm going to demand a big piece of this company, and then I'll make my money when you go public with an IPO, initial public offering, and then I'll sell my stocks and exit. I want 5-fold more than I put in if it works. If it doesn't work, I'm willing to take that risk. So Kleiner and Perkins said, yeah, you know, do you think you can make a new pharmaceutical company? There hasn't been a pharmaceutical company for 25 years. The barrier to entry is too great. Do you think you can do it? And Swanson said, yes, we have a new way. We're going to use living cells that have been genetically engineered to make the medicines, rather than the old way of mixing chemicals together in a tank and having those chemicals make medicine. Oh, well, that sounds interesting. Are you sure it's going to work? No. But you think it's going to work? Yes. And if it works, we'll make a lot of money? Yes. And so I'll give you some money. We gave money to these two smelly guys who were making computers in their garage, and you know that Apple turned out okay. So sure, I'll throw in some money. I'll take the chance. And I recommend this movie on venture capital. It is free on YouTube. It's called Something Ventured, and it examines this new funding model and the Traitorous Eight and Fairchild Semiconductors, and really what made the Silicon Valley the Silicon Valley. And remember, the Silicon Valley is also the Biotech Valley. So it isn't only, everyone focuses on the science, but it's really the business side also that came out of California. I've often thought you couldn't do this in Minnesota, right? They'd go, oh, I don't know, it sounds too risky. It wouldn't be prudent, don't you know? It doesn't have that cowboy, throw the Hail Mary, and let's take a chance on it kind of thing that California had. So that's the reason why biotech is here. Not only that the science was invented here, but also the business was invented here. And again, the city of South San Francisco, if you go there, there are cranes all up and down 101. Every single crane is a new biotech company. The last I looked, there were 250 biotech companies in the city of South San Francisco alone, and they are going into new areas. Let me talk about the traditional area, and by traditional I mean 40 years old, but then I'll talk about some of the new areas that are emerging, and in the end I would like to talk about how Modesto is a perfect place for one of the new areas in biotech. So here is Genentec Vacaville, and this is how I got into it. Genentec in 1994 said they were going to build the largest cell culture manufacturing facility in the world across the street from my campus in my county. So this is the manufacturing building. This building makes more money than Disneyland and Disney World combined. This building makes more money than the entire dairy industry in the state of California. This building makes more money than the NFL or the NBA or Major League Baseball. It makes a lot of money. I could do that all day. But part of the reason it makes antibodies, and part of that is that the antibodies are really effective. They're really effective at treating diseases for which, in the past, there were no treatments. And so I have sent, I don't know, hundreds of students across the street from us, and I tell them, at the end of the day, you are saving somebody's mom or somebody's son or somebody's brother. At the end of the day, that's what you're doing here. And this is the procedure. You start out with frozen cells that have been genetically engineered, and they're in a cell bank. You thaw those and begin them in a small tank. You feed them media, that is cell food. They divide before they're entirely packed in there. You move them to a larger tank, to a larger tank, to a larger tank. Ultimately, you trick them to make the protein that's coded for by the human gene that you've put inside of them, that someone else put inside. And then you separate the cells from the media. You then take the protein and isolate it to 99.99% purity using a series of isolating steps called chromatography. In the end, you sterile filter it, and you put it in a vial, label it, package it, and mail it to physicians for use on their patients. Remember that all biotech products are sterile. All of them are injected. The term is parenteral. You cannot take a protein pill because proteins function because of their particular three-dimensional structure. If you take a protein pill, they're going to unfold the minute that they enter the acid environment of the stomach. So all of these are injected. And to give you an idea, why do you need cells? Well, aspirin has 21 atoms, and an antibody has 25,000 atoms. And this slide is from Genentech. They compare the complexity of an antibody to aspirin. It's like a 747 compared to a bicycle. It's just that much more complex. It would be hard to make these in a tank, although there are people trying. And again, here is a protein. Proteins are the tools of the cell, and proteins can be used as enzymes. I already told you one use of that, tissue plasmid activator, a thrombolytic, an enzyme that digests blood clots. They're used for motion, structure. There are people that are making collagen. People that want collagen transplants currently are using animal collagen, often rats' tails. Now we can put the genes for collagen in a bacterial cell and have the bacteria make it under sterile conditions. And on the bottom, let me point out antibodies, and antibodies are going to dominate this field for the next decade. 5 of the top 10 drugs sold in the world are antibodies. So this is rapidly replacing aspirin and small molecules as the dominant form. And when you grow a giant tank, it may contain 100 trillion cells. Again, this slide is from Genentech. They compare that, that's 1,000 times more than the number of stars in the Milky Way galaxy. So let me show you some pictures. You can't take pictures in there, but Genentech, Vacaville was nice enough to put in professional photographers. So this is a tank, and it's growing mammalian cells. It grows a type of cell called a Chinese hamster ovary cell, and it's 3 stories tall. It has about the volume, if you can picture this, the same volume as the tanker truck that brings gasoline from the refinery to your gas station. Can you picture that volume? And again, it is going to fill with cells. The cells are going to make protein. The proteins are in that one tank are going to be worth on the order of $50 million. One tank, $50 million. Let me put that in perspective. A few years ago, we lost the entire crab industry in the state of California to a toxic phytoplankton. The entire crab industry in the state of California is $50 million. Entire industry, one tank. These are smaller tanks and showing some of the plumbing and the connectors. This is what you would look like if you were working in the plant. You're fully gowned. And the gowning is not to protect you from the product. The gowning is to protect the product from you. That is, all humans will give off skin cells. They'll give off hair, and you don't want that to get in. The isolation, the chromatography tanks, are on the right. They're 2 meters in diameter, and you can see a human on top, sort of blurry because he's moving, but that's for scale. That tank is the tank is filled with about $1 million worth of chromatography resin. Luckily, you can use it again and again and again. And then this is what it looks like, the automated machine that squirts a precise volume of your drug into the vial and then sterilely puts a sterile cap on it, puts a closure on it, and then ultimately labels it. So the scales are tremendous, but they're small compared to what's coming. Let me show you my campus. So we have a brand new building paid for by a bond. That is, the local taxpayers opted to tax themselves to add $10 per year onto their property taxes. It's something community colleges can do, but CSU cannot, and UC cannot. And so this is the inside of our building that was opened in 2017. These are bioreactors. Those are the tanks that are used to grow the cells under controlled condition. They detect and control the temperature, the pH, the dissolved oxygen concentration, so the cells encounter an optimum environment in order to grow at the maximum and produce at the maximum. This is our cell culture lab. I spent all day in this lab today, actually. What you see in the background are called biosafety cabinets. These are cabinets, areas that are designed to manipulate cells in a way where the bacteria that are on dust particles in the outside world can't get in and contaminate them. For mammalian cell culture, one bacteria gets into your experiment, you're done, you throw it away, and you start over. And we have a new certificate in synthetic biology, algal biotech, and we're working with Dave Menshew on algae, mammalian cell culture, and we just got a $3 million grant for stem cells. I'll show you that. Now, here is what's coming. So currently, biotech is devoted mainly to making new medicines. That $30 trillion bioeconomy that I was talking about is the vision of making everything using living cells, making all of the chemicals that are precursors to plastics. Rather than using petrochemicals, rather than taking it out of the ground, you use feedstocks, that is farm waste that's currently being wasted, and you use that as a source of sugars to feed your cells to make those products. And so if you think about everything that you're wearing, everything that you're driving, that is fuel, and think about a biological way to produce that, will reduce greenhouse gases and will build a circular bioeconomy that is not dependent on petrochemicals, that is not dependent on taking carbon out of the ground. Now, here is currently biotech, pharmaceutical biotech is mainly urban and mainly coastal. The next biotech will be rural. It will follow the feedstocks. And if you look at, here is a map of where the feedstocks are, and here is a map. The vision is that everyone participates in the new biotechnology. Everyone participates manufacturing new materials using living cells. Okay, so let's contrast pharmaceutical biomanufacturing to the new emerging bioindustrial manufacturing. In pharmaceutical manufacturing, you don't really worry about cost. Your number one consideration is compliance with the Food and Drug Administration's regulations. If you don't comply with FDA regulations, your product is adulterated, it is worth zero. So you can't cut corners because you need to comply with some of the strictest regulations in the United States. The regulation of drugs is probably second only to nuclear power and is comparable to the FAA regulation of aircraft repair. So let me ask you, you go to CVS and you take aspirin, and what's your confidence level that there's really aspirin in that bottle? There's aspirin. It says 50 milligrams per tablet. What's your confidence that it's 50 milligrams per tablet? Why? Why do you have such confidence? Because you know the FDA is very strict. Okay, now you're going to the vitamin store and you're getting a dietary supplement and it says ginkgo biloba. What's your confidence level? Very low. Why?
Not regulated.
Because the Congress explicitly took away FDA's power to regulate dietary supplements. And if you do the DNA analysis, about 1/3 of the herbs on the shelf have no trace of a DNA of the plant that's on the label. So again, as a comedian said, if you had milk and you had only a 2/3 chance that this cloudy white liquid is milk, you would hesitate putting those on your cornflakes, right? So the FDA, it's difficult to comply with FDA regulations, but it has this tremendous power of boosting consumer confidence. Every drug that you have has been tested 300 to 1,000 times for its identity and purity. Dietary supplements aren't tested. So you can't make this thing right. Now, that's pharmaceutical manufacturing. If you look at bioindustrial manufacturing, making commodity chemicals, making fibers for clothing, making, chemicals, citric acid or other chemicals for the chemical industry, cost is your number one priority. You're not making that much. Again, biotech products, the value, it might be $10,000 a gram, it might be $1 million a gram. In production of chemicals, it might be $1 a pound. If you're making fuel, what's your target? If you're making biofuel, what's your target? Your target is higher if you're in California, right? You want to be $4 a gallon, right? If you're making chicken nuggets using chicken cells in a tank, what's your target? Whatever the cost of chicken is, you might have a premium because now you can have vegan chicken, but you know that cost is everything. What's the cost of your carbon? What's the cost of your process? And it's this differential in the value of the product. So the idea is to build a new bioeconomy that is comparable in scale to the current size of the petrochemical industry. Comparable in scale to the current size of agriculture. What does that mean for the young people in the world? What does that mean for the young people in this room? That means that there are going to be career opportunities that start at a livable wage and move up from there. So my students start, without an associate's degree, my students start at $25 an hour. That's $50,000 a year. But what the companies offer them is an ability to work hard and then move up, move up, move up, move up. So they make a lot more than we make. And I went to college for 12 years. So within a couple of years. So anyway, that is another byproduct of this industry, providing opportunities in a career, not a job, but a career that provides a livable wage. Okay, so this is actually my son. He went through our biotech program and then got his bachelor's degrees at UC Santa Cruz. And there's a funny story. Before I got my job at Solano College, I taught at UC Santa Cruz. So when he was born, I was wearing a sweatshirt that said UC Santa Cruz Biology. 22 years later, he graduated from UC Santa Cruz in biology. It was preordained. Then he went to the University of Edinburgh and got a master's in biotechnology. So you can't push your children, right? If you push them, they're going to go, oh, I'm going to be an art major. So you just, hey, let me show you something interesting, and maybe you'll wander your way in there. And anyway, he's currently working for a biotech company that is growing chicken in a tank and wants to make chicken without chickens. It's chicken cells, so it is chicken, but without chickens. He's working for Upside Foods. But I wanted to point out one of the emerging areas of biotechnology, and this is cell therapy. The most common type of cell therapy right now, and there are 6 approved products, is CAR T therapy, chimeric antigen receptor T cell therapy. This is basically a cure for cancer. For the first time, physicians are using the C word, cure. 10 years ago, they would have said, cancer is something that you manage. It's like diabetes. You manage it and you keep it from killing you until something else kills you. Now, for the first time, they're talking cure. And let me take you through this. What you do is you take a patient, and that patient is a month away from death. They have tumors all over the place. They were treated with chemotherapy, and the tumor evolved its way around. And then you hit them with different chemotherapy, the tumor comes back. So you're really at the end, and what you do is the physicians remove their T cells, remove their immune cells, their white blood cells, and then mail them to a cell therapy company. It might be Bristol Myers Squibb in Seattle. It might be Kite in Southern California. And that company takes those cells and uses a derivative of HIV, a lentavirus, uses a virus to introduce a new gene into that cell, a gene that codes for a protein that the patient's cells are going to display on their surface that is specific for that cancer. You activate them, grow them up, and then mail them back to the patient. You're on the clock because the patient only has a month, so you have to do this in 2 weeks. Mail them back to the patient. The physician puts the cells back in the patient. It goes around the body, and the patient is tumor-free in 2 weeks. Certain death to tumor-free indefinitely. And it works sort of 90% of the time. What's the catch, ask me? $500,000. Okay, so if everyone gets this, it could break healthcare. I don't know, maybe healthcare needs to be broken, but that's another thing. Here's gene therapy. Here's 2 of my students. And gene therapy, you have a child who was born with a genetic disorder, inherited the wrong genes, that is, inherited genes with the wrong sequence, usually from both parents. And without treatment, they are going to maybe depending on what their condition is, have a slow, degenerative future marching toward an inevitable death. And I've spent a lot of time at Oakland Children's Hospital. It's really the center in Northern California for genetic disorders. I've talked to a lot of parents. Again, I can't tell you the parents know where the research is. The parents know where the clinical trials is. The parents are hoping that this breakthrough is coming before that end occurs. So what a company will do is take that gene and take a harmless virus, take the DNA out of that virus and insert the missing gene, grow up a bunch of it, purify it, inject it into the patient. The virus circulates through the body, and there are, for those of you who are mathematicians, a dose is 10 to the 15th viruses, one with 15 zeros behind it, and all of those viruses go, they find the right organ, and the virus then is a tool to insert the DNA, to inject the DNA into these cells. That DNA supplies the function of the missing gene, curing that disease. What we're really talking about is the blind will see, the deaf will hear, the disabled will walk. What's the catch? 2.1 million for one treatment. Another one was just approved for 3 million. Okay, so the price will come down, but it's a cure. And again, think of the cost of not treating. Think of the cost of a kid who's in the hospital a couple times a month. And then, as they slowly get worse and worse, home care, 24-hour home care, going into hospice, you're chewing through healthcare dollars, and it might be that you're saving money at $3 million. So, but are we really talking money? We're talking children. We're talking alleviating human suffering. But we have to figure out a way to pay for it. Good. So Dave is involved in these organizations. These are national organizations paid for by the taxpayers in order to spur innovation and to keep the United States competitive in this area, to keep the United States dominant in this area. Let me mention another innovation, the 2020 Nobel Prize. And this is, again, for Jennifer Doudna. This is a local story. She's a professor at UC Berkeley. This is CRISPR. This is an editing technique, a gene editing technique that is like it's like a cut and paste in your word processing program. You can go in and fix the broken part of the gene. And I was reading today about several clinical trials that are being performed on children with sickle cell anemia. Someone with sickle cell anemia has a single base pair different, and it creates episodes once a month of intense pain and life-threatening, and again, tremendous cost, but this looks like, again, a cure for genetic disorders. I wanted to show you something fun. What we do a lot, and in science we use a protein called green fluorescent protein. It is part of the bioluminescent apparatus of a particular jellyfish that is native to the coast off of Washington State. And what you do is you take the gene for that protein and inject it into bacteria that now can glow green, or inject it into mice that now can glow green. You can buy fish that have not only that gene in it, but fish that may have red fluorescent protein or yellow fluorescent protein. You can currently get these at Walmart. I'd be willing to guess that your local Walmart sells these. And you can inject it at the unicellular stage, a fertilized egg cell in a rhesus monkey, and get it to glow green. If you can do it with a rhesus monkey, can you do it with a human? For sure, yeah. I don't know, do you want to glow green? I'm thinking genetic tattoos, right? The other thing I want to do is I want Christmas trees. Yeah? More than that. Okay, so this is a brain tissue, and it has different cells turn on different fluorescent proteins. And so rather than a rainbow, this is a brain bow. And you can tell where the cells are. And here's a skin bow. So why would you want to do this? Well, where this is really useful is if you wound the fish and watch where the cells come from that repair the wound. For such an important function of medicine, we know relatively little about wound healing, and so that allows you to do this. But here, I put some money into this Kickstarter program, and they were trying to put lightning bug genes into plants to make them glow, just like Avatar. But this one didn't work. But in April, I went to the big Biotech conference in Oakland, and somebody had put fungal bioluminescent genes in petunias, and they glowed really well. So the idea is to have plants on the side of the road that are glowing green rather than street lights. I kind of like it. Here, these are littermates. The one on the right had human growth hormone injected in, the gene for human growth hormone injected into it as a one-cell embryo, and now we made Mega Mouse. These two salmon are the same age, so what you can do, you have a genetically engineered salmon that grows much faster and comes to market earlier than it would without that genetic engineered. So you can genetically engineer goats to secrete spider silk in their milk, and so there you have spider goat. Gene therapy we talked about. The Human Genome Project, this is an interesting story. It has sort of a hero and a villain that many books have been written about the race to get the Human Genome Project. Craig Venter started a company, and he wanted to beat the government at its own game. Francis Collins was in charge of the government end of it and wanted to get all 3 billion ATs, Gs, and Cs of the human genome and publish it on the internet for everyone to use. Craig Venter wanted to patent everything that he had, and in the end, they all got together and declared a tie. President Clinton got his science office. He said, get those guys together, and they met over pizza and declared a tie in February 2001. And now you can have your genome sequenced for under $100. And so everyone's going to take their genome and take it on a flash drive, take it to their physician, and their physician's going to look through and say, oh, look, you have this gene, that drug won't work on you. Oh, I'm going to stay away from that drug, you have an allergy to it. Oh, you're a perfect genetic candidate for this drug. Now, imagine how much money you're going to save because you're not trying drugs that aren't going to work. You have this pre-screening, and you're only going to use the effective drugs on that. So that's going to save money. Many of you probably have seen 23andMe, and that will give you your ancestry, and it will give you some genetic predispositions. The problem with this is we only know about bad news. You're not going to get any good news usually. We only know about diseases. You're not going to, you won't get any good news. This still sounds like science fiction to have a test tube baby, but the test tube baby is having test tube babies. So Robert Edwards was the first one to successfully implant an embryo that had been created not in a test tube, but in a cell culture plate. And now there are on the order of 5 million people walking the planet whose origin was not inside their mother. And this creates some interesting ethical issues. So a woman who doesn't want to really get pregnant can hire somebody. What's the going cost? $10,000. So you hire someone to carry your baby. What's been happening a lot is a woman who has a uterus that can't carry to term will donate the egg, and then her mother will carry it. So then her mother gives birth to her own granddaughter. And the oldest I've heard so far is a 66-year-old mother gave birth to her granddaughter. Now, tell that to your grandparents, right? I mean, we're there. Stem cells are huge. Stem cells, half of you in this room will be treated with stem cells. It's a silly thing. The most common operation in the United States is a knee replacement operation. You're putting in metal and plastic parts into a knee when there's just a single layer of damaged cells. Grow those cells and then implant them. And I'm teaching a stem cell class. What's really fun is one of my students, I first met her when she was in a wheelchair. She had been treated with stem cells for her knee damage. So she's a stem cell student who had been treated with stem cells. And artificial organs, you can grow them in rats for some other reason. About 80 people have been implanted with artificial bladders. It's pretty easy to make the bladder. It's hard to hook it up to the neurons to send the signal, hey, I've got to pee. So anyway, they're still a work in progress. This is a heart that you take a heart, remove the cells, and leave the protein scaffolding, and then use stem cells to repopulate the heart. The idea is to do this with pig's heart, which are abundant, and then repopulate them with human hearts, and now you no longer have a shortage of hearts for people that need a heart transplant. And that's the idea. 3D printing of tissues, so you can print ears, and you combine it with electronics. Again, the deaf will hear. So thank you, taxpayers of California. You voted in 2020 to tax yourself and to put $5.5 billion to keep California dominant in the field of stem cells. And this is where my college's, Solano College's $3 million in grant money comes from. But this is going to lead to more of those cell therapies, CAR T that are cures for cancer, and also gene therapies. This is going to fuel this revolution in the state of California. And again, there's the California Institute for Regenerative Medicine. You've probably heard about Dolly. Dolly was the first mammal that was the result of a nuclear transplantation. You took one breed, put it into the egg of a different breed, allowed that to divide in a cell culture plate, and then put that into a different breed. And which breed is the lamb that's born? The breed of the first one, of the nuclear donor. And I was at the Chicago Museum of Science and Industry, and they had 3 mice, and they had the breed of the nuclear donor, the breed of the egg donor, and the breed of the surrogate. And of course, they had the cloned mice, and the cloned mice are the same breed, the same color as the mice that donated the nucleus. Now, why would you want to do this? Well, what you can do is take somebody's nucleus from an adult cell, put it into an egg, have that egg divide, produce stem cells, and now that stem cell is that person's cells, which you can make an organ and it won't be rejected. It won't be rejected because it's the tissue type of the person who donated the nucleus. GMO crops, but that is golden rice, which has the same molecule that carrots have, which are good for eyesight, and that way you can supplement them. Here's something fun. This is a Bay Area company. I love this company. It's called Perfect Day Foods. They take genetically engineered fungi, they feed them sugars, and then the genetically engineered fungi make milk protein, so now you have milk without cows. And so they want you to know that without cows you have a smaller hoof print. And if you want to work for them, you join the movement. Anyway, I just had Brave Robot ice cream made with this. It tastes like ice cream, and this is milk without cows. The Chinese government is very interested in this because they want the nutrition of milk but don't want to cover the entire countryside with cow poop. You probably have had an Impossible Whopper. This is also a Bay Area story. Here you take a genetically engineered bacterium and it makes a vegetable protein that's similar to hemoglobin, and therefore your plant protein your plant patty bleeds. Here's bacon made by fungi. This is a silk tie made by spider silk. The bacteria have been genetically engineered with spider genes in order to make spider silk. It's currently about $150. The same company makes artificial leather using fungus. And there are many, many companies that are looking for an alternative to leather. Synthetic life, you can now take chemicals off the shelf and create a living cell. And this is the first time that this was done. That's Craig Venter running the Craig Venter Institute, and here he is getting the presidential medal from President Obama. Algae, we're working on algae. You can see how the productivity of algae. You can grow algae in a tank. You can feed it as well as grow it through photosynthesis and trick it to make a company, to make a product. The Algae Foundation, that's what Dave Menchu and I are involved in. And CalCab, the California Center for Algae Biotechnology. I like their logo. You like the algae smiling face? Somebody has a sense of humor. You can make oil from algae. It's really good as jet fuel. It has the right properties for jet fuel. It isn't clear that you have to age. You can turn off the aging genes. Aging is probably more a programmed self-destruction than it is wear and tear. If you could turn off the aging genes, we would all be like the elves in Lord of the Rings and not age. Of course, you have to deal with the societal consequence of that. And from the beginning, the Human Genome Project devoted 3% of its budget to ethical, legal, and social issues. They knew they were going to disrupt society with the great power of genetics. And there is a fellow in China who is sitting in jail because he CRISPR'd 2 twins and really not only did the world, but more importantly, the Chinese government came down on him. I recommend the movie Gattaca. It moves really slowly, but it examines these ethical issues. The other thing I like is that Ethan Hawke and Uma Thurman met on the set and they had genetic experiments of their own later. Yeah. Good, and then biohacking. I recommend the Netflix 4-episode series, Unnatural Selection. I have a small cameo, look for me in episode 2. And the Odin is a company that sells kits. I just ordered a couple so you can do genetic engineering in your garage. This is one kit they have. They have the small frog, and you can genetically engineer them to have a gene with growth hormone and make bigger frogs. What do you think of the ethics of that? It doesn't matter. They're bad boys. They're hackers. You can probably terraform Mars. Again, I recommend the LA Times articles on coronavirus and on making the vaccine. They are spectacularly well written. They don't dumb it down, and they're really good. And so I'll give these links to the organizers. Here are my students, and just to show you what a day in the life looks like. This is Nathan. He's a former member of the United States Marine Corps. I don't know if you can see it. He's 6'8" and is of Scottish descent and throws the caber, tosses the caber. I can't even lift the caber, but his former specialty was weapons of mass destruction. Here's our team. And then let me talk about what we put together across the country. And what we have at Solano College is we have multiple certificates, so you can get a job really with 4 classes. And then, you could transfer from here or find a more local college. And then an associate's degree after you add after you add general education. And then seamlessly into our bachelor's degree. Our bachelor's degree beginning and 4 years under $10,000. And then we've negotiated with 6 different graduate schools to accept our graduates. And we have outreach to high schools, including Dave's students. And so we have a pathway from high school through PhD with multiple entry and exit points, depending on what your family circumstances are. Again, 100% placement because the field is so hot at every exit point, and people can jump in and jump out depending on what their family circumstance is. And we have a summer boot camp where we compress our entire program into 8 to 5, Monday through Friday for 10 weeks. And again, there's our labs. And here's something fun. With my teaching job, sometimes students meet in biotech, and so they get married. Here's another one. And more than that, they asked me to perform the Dearly Beloved. We're gathered here in the presence of these nerds. Because, and here I am doing it again. I've done it 4 times. So when I got hired with my teaching job, there's this little phrase at the end. It says, and other duties as required. I didn't know that this might be one of the duties. So anyway, it's really fun. And let me end with telling you why I'm such an advocate of biotechnology. So this is my son when he was 4. And on Thanksgiving, he fell really ill. He had 102 fever, but that wasn't what was unnerving. He was delirious. And he was telling a story, and I said, did that happen in your dream? Did that happen yesterday? And he just didn't make any sense. And I asked my wife, it's 102 fever. Do we take him into the emergency room? Because you don't want him to think that you're a hypochondriac. And I said, what do your instincts say? And she said, my instincts say take him in. Then let's take him in. Had we not taken him in, he would have been dead in 4 hours. So we took him into the emergency room, to the local emergency room, and this rash started to form. And his mother said, what's that rash? And the attending physician took one look at it. He said, I need a helicopter. I need an airlift here in 5 minutes. I need airlift in 5 minutes. And he said, I'm sending you to Oakland Children's Hospital. So he takes off, and then we drive down the freeway to Oakland, and I get to the front, and I get to the front desk, and I said, I'm Jim DeKloe, Jefferson DeKloe's father. And they said, oh, ICU. ICU? It was 102 fever. Although they did airlift him. And then I get to ICU, and it's a security door, and I pick up the phone. I'm Jim DeKloe, Jefferson DeKloe's father. Oh, yes, we've been waiting for you. A social worker will be out to speak with you. I want the doctor. I didn't say that. But the social worker came out and said, he's really sick. You've never seen a kid this sick. You're not going home today. You're not going home this week. You're not going home this month. He's in for the fight of his life. I want you to prepare yourself. It's a little frantic back there in ICU. They've got a team of about 12 working on him. They're pumping in 17 different drugs through 3 deep lines, and I'll come and get to you when it calms down. So after a while, I'm sitting there going, what? They come out and get us, and the attending physician says, here's how it is, Dad. We'll try and save him. We'll know today. I have to tell you that survivors usually have all of their limbs amputated, and brain function we're going to worry about in a month or 2. Do you have any questions? Let me process this for a minute. And then I got the diagnosis. It's meningococcemia. So meningococcemia is a disease caused by the bacteria that's the most common type of bacteria that causes meningitis when it gets into the fluid of the central nervous system. In this case, it jumps in the blood, and it's nearly always fatal. It causes endotoxic shock, and in his case, it caused every clotting factor in his body to go off. So that's what that rash was, and when I saw him, if you think of the deepest bruise you've ever had, he was that over 100% of his body, except his fingers and toes were turning necrotic black. And so we were in trouble. And then they said something. So, well, I called my father-in-law, a pediatrician. He said, Jim, I got to tell you, you're going to read about a survival rate. There's no survival rate. I've seen a lot of these cases. I've never seen one survive, not one. So he hung up with me and he calls the family and he said, I'm going up for the funeral. I'll call you when it's over. So he starts to drive up. And anyway, so then the physician says to me, we'd like to try something. We've tried it once before. Do you see this coming? He has blood clots. My favorite drug. So, right, I said, a thrombolytic, you have to give him TPA. And they said, you know about TPA? I worked at Genentec on TPA. That's my drug. I built my entire program around TPA. There's no drug in the world I know more than TPA. You've got to give him TPA. He's got blood clots. You have to digest the blood clots. And he said, well, we have to tell you the possible risks and the possible side effects. I can tell you the risks and the side effects. We have to give him TPA. And the risks are that if there is a puncture in a vein in the brain, if there's an aneurysm, and you give him TPA and it's being held back by a blood clot, then it's going to digest that blood clot and he's going to bleed out. But what I knew TPA had a half-life of 1.5 minutes. So I knew that if something didn't happen in 1.5 minutes, we're probably okay. Anyway, they dripped in TPA and it digested all the blood clots and saved him from certain death. So of all the things, you couldn't invent this for Hollywood, right? Not only a Genentech product, the company I'd been working with and for, the drug that I'd worked on, and saved him from certain death. And there he is on his way to medical school. He's a third-year medical student. So again, if you want an advocate for this field, you can find no better advocate than me. He would have been dead that day. I'll end with one other funny story. So he was in a coma for a long time, and we had 56 operations. That's a real number. That's probably why he's a doctor, or studying to be a doctor. But he was in a coma for a month and a half, for a month. And so blood stopped in his body, right? And so the question is, does he have any brain function left? And I thought we were okay because he wasn't drooping or asymmetrical, but so they're going to take him out of his coma, and they pulled out all the lines, and he's on his back, and when you're on your back for a month, the mucus kind of sort of sits there in the back, and so the nurse is going in with suction, trying to get some of the mucus out. And he's coming around, and you can kind of see that it's uncomfortable. And so it's building up to this dramatic, is there any brain function? And all of a sudden he says, stop that, do that to my brother. And I go, okay, we're okay. The sibling rivalry is higher function. Okay, we're okay, and that broke the tension. But again, he's here today because of this field. And it is the field of now, and it is the field of the future. Let me really close by saying, on the way down, I was talking to my friend who worked in Modesto for a long time. And he said, Jim, what you're going to encounter in Modesto is it's a very tight community. It is a community that takes care of itself, and it's a can-do community. So he did a project here, and he just loved being here. And I think that you have all the tools for this next generation of biotechnology. You have one of the biggest fermenters in the world close by, and you probably know people that work there, and the science there is spectacular. They have spectacular scientists there. And also, you have an ethanol producer a little farther south. You have feedstock. The next generation is going to use plant waste and like orchard waste. And of course, I've heard there's a little bit of cow manure from the dairy industry, and that can be used for anaerobic fermentation. And so really there are all the tools here for this next journey in a $30 trillion bioeconomy that for the young people, this is a place that you can have a career with a livable wage and upward mobility. But more than that, a lot has been said about this upcoming generation. What I've found to be true is the upcoming generation doesn't merely want to pick up a paycheck, but wants to make, as Steve Jobs said, a ding in the universe. And this is where you could do that. And let me close there. And again, thank you for inviting me. And thank you for your attention. So I know, did I go over, or I'd entertain some questions if you'd like, or I can hang out after if anyone would like to talk to me after.